Okay, in this last section of the chapter, we're going to be looking at some of the ways that these microbes persist and some of the pathologic conditions they cause. And we need to remember that a lot of times a microbe can be dormant, not all of them, but some of them, and these are scary because you have them and you don't know you have them. But we're going to look at what reservoirs are. A reservoir is the habitat of the microbe where it's stored and spread. And so we're going to look at different types of reservoirs. The first type is the living reservoir. And here we look at carriers, which is an individual that has this pathogen and spreads it around. A lot of times they're asymptomatic. A lot of times it's where the microbe incubates and then they carry it further. Then we have, we also have animals that are reservoirs. And we talk here about vectors, an animal that transmits that infectious agent from one host to another. And most of the time these are arthropods. In other words, most of them are going to be insects. And we look here at the biological vectors that actively participate in the pathogen's life cycle. It would cause signs and symptoms in the biological vector. And here we're looking at the mosquitoes that transmit malaria. The mechanical vectors transport only the infectious agent, usually on the outside of their body. And here we see the fly on the right side that would walk around in the feces or whatever and possibly transmit cholera that way, because it would just fall off of its body. Now here's another vocab term we need to be familiar with. It's zoonotic. These are animals that an infection that's indigenous to the animals that can be spread to humans. And we hear a lot about bird flu. It started out in birds and it's now transmittable to humans. Swine flu is another example of a zoonotic organism. Here, figure 13.9 in the old book, 13.10 in the new one, are some common zoonotic infections and how they are spread, the animal reservoirs for them that you'll just look over. Then we have the non-living reservoirs, and of course we come in contact with soil and water all the time, and we know that there are a lot of microbes that are in the soil and water. Some other vocab terms: whether a disease is communicable enough, can the infected host transmit it to another host so that it gets infected, or is it non-communicable? Is it acquired through some other way? Accidental contact, like maybe you use a towel in the restroom that has a microbe on it and it causes you to have problems and get sick. Now, transmission can be contact transmission or indirect. There's always going to be some way it's transmitted by some vehicle. It can be a contaminated object like doorknobs and telephones, or it could be just the air, because in the air sometimes people sneeze and so forth, or just in the air. A lot of different things can be transmitted that way. Now, direct contact would be touching, kissing, and intercourse. In the last part of this chapter, we look at nosocomial infections. These are infections that you get while you're in the hospital. And that's why asepsis is so important. These are opportunistic pathogens that are going to sneak in when the immune system is compromised or there's been some sort of invasive procedure, and they're going to use that opportunity to establish themselves and cause some secondary type infection. That's why asepsis is practiced in our healthcare so much. We also use in healthcare universal precautions. That is, we assume that all patient specimens have infectious agents in them. And we do the same thing here with our microbiology lab. We assume that everything that we work with is infectious, and we treat it that way. The last thing that's mentioned in your chapter is determining what's the causative agent, what's the etiological agent. And Robert Koch developed these postulates which is the standard for determining what causes the disease. You may remember him from that scientist crossword that you did the very first of the semester. But you do need to know these 4 postulates. First, you got to find evidence of that microbe in the case of a disease. Then you isolate it, and then you inoculate a healthy specimen with it and observe to see if you get that disease, and then you re-isolate the agent from the subject. But you do have to know these 4 postulates and be familiar with them.